Good day, everyone. My name is Jane Natividad, and a scientist at Nestle. Today, it is a pleasure to present to you the research work that we conducted to understand the ability of human milk oligosaccharides, which I will refer to as HMO, to protect against inflammation-induced epithelial barrier dysfunction in vitro. The presentation is part of a larger study in which we investigate the overall impact of HMO on epithelial barrier function. We have another webinar presentation that talks about direct effects of HMO on epithelial barrier, and I would highly encourage you to similarly watch the presentation. I would like to begin the presentation by giving a little bit of background on a key role of intestinal epithelial barrier and knowledge of the effect of HMO in the intestine. Dysregulated or leaky barrier have been recognized to be an influential factor in the initiation as well as progression of plethora of diseases, including intestinal inflammatory diseases, allergy, as well as the metabolic disorders across lifespan. Just shortly after birth, the gut epithelial barrier is so much looser, and within months after birth, this becomes tighter. At a certain point, both breastfed babies and formula-fed babies have similar epithelial barrier tightness. However, studies have demonstrated that breastfed infants have faster evolution towards tighter barrier than formula-fed infants. This is associated with higher protection of breastfed babies against inflammatory and metabolic diseases. The protective effects of breastfeeding have been attributed to the nutritional bioactive molecules present in the human milk. Among them are a group of human milk oligosaccharides or HMOs with different chemical properties. The direct effect of HMOs on epithelial barrier have been demonstrated previously by other groups. Specifically, intact HMOs are able to modulate the proliferation and differentiation of intestinal epithelial cells. Additionally, HMOs are fermented by intestinal microbiota in the colon. The product of microbiota fermentation generates functional metabolites, which may have gut epithelial barrier benefits. As I mentioned in the beginning, we're also evaluated the indirect effects of HMO through microbiota fermentation on epithelial barrier. So please free to watch the webinar if you're interested to know more about the results. Overall, this gives a reason to believe that HMOs may contribute to the proper development of the intestinal barrier in breastfed infants and subsequently protection against diseases during early life up to adulthood. We globally hypothesize that HMO can protect the intestinal epithelial barrier against inflammatory challenge both directly and through the fermentation products. We further hypothesize that different HMOs, due to the virtue of their chemical composition, will have differential capacity to impact the barrier. This is clinically relevant as the intestinal lining is in constant challenge with the environment, whether from infection or food antigens. And the ability to protect the barrier can prevent complications and help in maintenance of intestinal homeostasis and overall health. For this presentation, I will focus on showing you our study to determine the different effects of HMOs. This study has two tiered objectives. First, to determine whether a combination of 6 intact HMO, which I will refer to as HMO6, is able to strengthen the barrier prior and during inflammatory challenge. The 6 HMOs to be tested are 2FL, 3SL, 6SL, LNT, LNNT, and DIFL. They were chosen as they are the most abundant chemical structure naturally present in human milk. Second objective is to identify among the 6 HMO which one predominantly drives barrier effects. To answer our objectives, we use a co-culture in vitro model of 2 cell lines, one of which is able to produce mucus. Hence, the model is able to capitulate not only the absorptive properties of epithelial cells, but also the mucus layer component. The cells are then grown in vitro, and during the experiment, the cells were first pre-treated with HMO, with lactose, which we use as control, or vehicle, which is the solution used to dissolve HMO for 48 hours. We then challenged the system using classic cytokine inflammatory challenge for an additional 48 hours. Throughout the experiment, we measured transepithelial resistance, or TER, which is measurement of permeability of ions across epithelium. Furthermore, at the end of the experiment, we measured permeability of macromolecules from the apical to basolateral membrane using a permeability marker called Fitz-detection. I will refer to this method as FT4 translocation in the following slides. This method represents translocation of food antigens and bacterial products that may be detrimental on the health of epithelial barrier. For both of the objectives, similar experimental protocol were implemented, with the first study looking at HMO dose responses. So moving on to the results, we first look at the effect of HMO prior to the challenge. As demonstrated on this slide, so if I can allow you to focus your attention on the figure at the left side, you can see that HMO-treated cells show significantly increased transepithelial resistance regardless of dosage compared to controls. After the challenge, barrier leakiness was decreased by HMO-6 treatment, as demonstrated by lower transepithelial resistance and FT4 translocation compared to control-positive cells, which are cells stimulated with the cytokines. At higher dose, you can see that the transepithelial resistance and FT4 translocation are comparable to control negative, which was not stimulated with cytokines. Furthermore, the effect of HMO-6 seems to be dose-dependent. We then ranked the efficacy of HMO at similar concentration. As demonstrated in this slide, different HMO have differential effects on FT4 translocation, and 2FL followed by LNT seem to be the most efficacious in protecting the barrier integrity, while lactose control did not significantly reduce FT4 translocation compared to control-positive cells. Overall, the blend of 6 intact HMOs reinforced epithelial barrier and prevented the effects of inflammatory cytokines on the barrier in vitro. Among the HMOs, the presence of 2FL and LNT in HMO-6 blend seems to contribute substantially to its protective effects. Finally, our results confirm previous reports as well as support a key role of intact HMOs on the promotion of gut health and on the protection against intestinal inflammatory disorders by human milk. Thank you for your attention.